it going?
Yeah.
All right.
We're good.
Okay, we're good. All righty. So our guest speaker today is a clinical lab scientist, also known as a medical lab scientist. This happens to be my nephew. His name's Quinn Bates. And he graduated here from Weber High back in 2001. So a little bit of what's 20-year reunion coming up? Oh my gosh, that seems so old. Anyway, Quinn came through here. He was involved in football, he was in the chamber choir. Do we have any choir members in here? Anyway, and he actually had my class. It was kind of weird to have your aunt as a teacher.
Yeah.
Kind of odd. He sat just right back over there where JC, actually in front of where JC sits. So he had to muddle his way through my class, and then he went up to Weber State and went on to bigger and better things. So let's thank him, because he works at the Altaview Hospital, which is way down in Sandy, and he actually lives in Harriman. So he drove an hour and a half to come and share his career with us. So let's thank him for coming.
Awesome, thanks for having me. Okay, so like Ms. Rado said, went here. Then I ended up going up to Weber State. That's where I actually got my bachelor's degree. And super fun. I did art while I was here, loved every minute of it, and super fun. So, how many of you know exactly what you want to do when you're done with school?
I want to go into sports medication.
Sports medication. Anyone know anything else? And it doesn't have to be necessarily medical.
I want to be a nurse practitioner.
Nurse practitioner? Awesome. How about something outside of medicine? Do you guys know pilots or anything like that?
I know degree and getting on a job, maybe a business degree.
A business degree? Perfect. So when I was sitting right there in that chair, I knew 100% that I was going to be an artist. And what happened was, I knew Leonardo da Vinci learned anatomy and physiology so he could draw the body better, right? And I was like, hey, if it's good enough for him, it should be good enough for me. And that kind of opened the pathway to kind of blend art and medicine together. And it set me up for a really good career. So with that being said, I think that we should really actually get to know each other a little bit better. And I think the best way to do that, actually I know the best way to do that, is to swap some fluids. So let's do it. Come on up. No, I'm serious. Like, come up here, right here. I got these fluids. And I want you to grab a vial of fluid and a pipette. Come on, let's go.
What?
That's what I thought too. We'll never know. I don't know.
Yeah, just grab a tube, grab a pipette, and back.
Hey, Ethan, when we're finished with this part, can I get you the this? Oh, like in a different chair. Thank you. Because I'm just getting like your whole torso in and stuff.
So where's that about? Right about here.
Yeah, and then when you do your PowerPoint, I'll just pan over.
I just want to know where I need to be.
Yeah. Yeah, we're getting the full screen in, so you're okay.
Okay, perfect. Okay, so this is really simple. All you're going to do is you take up a little bit of your fluid, have your neighbor do the same. You're going to take some of your friend's fluid. You're going to swap fluids. Okay? Make sure you leave enough in there for I'll get some back. Okay? Okay. Now, you guys are like really adorable. I want you guys to like move like these people over here, those people over there. Swap some fluids. Find your neighbors.
Drew, you want some fluid?
Yes. There you go.
Don't be shy. Swap some fluids.
Monica.
Okay, go find like 2 more people to swap fluids with.
2 shots of vodka. Well, you don't have to.
Yeah, no, I'm putting it back.
Monica, I just got that on film. I don't know if I'd be saying that.
I haven't swapped it yet.
Okay, finish up whoever you're swapping fluids with. Find your seat again. Okay. Awesome. So, let's talk about, like, biologically, like, what kind of fluid could this possibly be? Well, I mean, I'm a medical laboratory scientist, so what kind of body fluid could this be? Saliva, that's a good one. You guys think of anything else in the body? Tears. No, that's actually great. Sweat. Yes. Okay. Anything else that you guys can think of? Okay. Cerebrospinal fluid looks exactly like this. Does like 80% of your glucose, your brain works really hard. Your guys' brain working really hard. Okay. Maybe urine if you're really hydrated. Maybe blood if you're on IVs. You know what I mean? Different things like that. So let's stick with the saliva idea. Okay. So obviously you know what I've done to you, right? I contaminated you. So what I need you to do is take some of this, and you want to basically, you know, double it. If it stays blue, you're okay. If you turn pink, however, you're not so lucky. You've been contaminated. Okay. Patient 0. What'd you do?
She has the coronavirus.
Oh,
me
Yeah.
Nuts.
That means I'm contaminated.
Yeah, double it.
Oh, okay.
Yeah, go nuts.
Cheers.
Oh,
Coronavirus.
Ooh.
Oh, you
Go ahead and
guys.
pass that.
Ah, I bet it was hated.
Now, I would agree, isn't swapping fluids super fun?
But
But I had a lot, so.
Yeah, but you swap the same.
Yeah, but we can tell that it stayed blue, so you're safe. You just look at that top surface and see that it stays the same.
I like this man a lot.
You guys are good.
Did you change pink?
Just whiskey.
Yeah, give it like a whole squirt.
Oh, yeah. I'm like purple.
Yeah. Mine's purple.
Mine's like, what's the purple? Mine's like purple.
Yours is pink. That's pink, brother.
Oh, yeah. Oh, yeah.
Wow.
Mine's
Okay,
yellow.
has everyone had a chance to get the
See, mine's yellow.
Smurf juice? No Smurfs were harmed in the creation of this juice. Okay, so here's a quick question. Okay, all the students who have pink tubes, stand up. Wow, look at that. Wow. Okay, perfect. Got it. Over 50%. Okay. So if this was a clear fluid, we call it saliva, right? So what kind of things could be transmitted by swapping spit? Mono. It's got another name. Also a common name.
Kissing disease.
Exactly, kissing disease. So this is part of my field. To look at disease and how like it spreads and the treatment and identification of it, okay? So I actually got mono in college, but that's another story. We'll pass this around, and I actually gave it to my wife. So,
You guys can put your droppers just right down inside the
yeah,
desk.
go ahead and set the whole thing in there. So what I'm going to do right now is, as we're passing this around, I'm going to put on a a short video about the very first facility that I worked for and worked at, to kind of get you acclimated to what a medical laboratory scientist does, and kind of see like the actual environment in which I worked. Okay.
It's fascinating that a single drop of blood holds the key to healing some of the sickest people. We're here at Intermountain's Central Lab in Murray, one of the most sophisticated medical laboratories in the country. Think of this facility as a great detective agency where a team of specialists tries to uncover all kinds of information locked inside body fluids and tissue cells.
The biggest improvement I've seen from 10 years ago would be that we get much quicker turnaround time with our labs, including even our specialty labs such as therapeutic drug levels. That's a huge advantage with our complicated patients where we're constantly monitoring and adjusting their medications. We also are able to access the labs so quickly with Intermountain's computer system, and that's a huge advantage for us. A patient can have blood drawn and come to clinic, and their labs are already available and able for us to access instantly. We're unable to do our job without the central lab and the services they provide. I can't evaluate a patient, especially a liver patient, without their labs and without their biopsies. I would have no idea clinically what's going on without those services.
So what goes on inside the Central Lab? Here's David Gardner, COO, to give us a behind-the-scenes look.
Well, it's hard to paint a complete picture of everything we do here at the Central Lab because of the hundreds of tests we do. We do things from ammonia testing all the way to vitamin D, from blood cultures all the way to flu testing. Let me show you. In fact, right here is our automated area. This is our lab where we run about 90,000 specimens a month, where we call it our chemistry and hematology. And in this area, we do things like test for your blood calcium, glucose levels, white cell counts, and those types of things. Over here on my right here is the molecular pathology department, where we test for viruses and other types of things. In fact, during the H1N1 season, we tested over 35,000 specimens during that flu season. We use a technology called PCR, or polymerase chain reaction, where we actually take a sample, amplify it up, and look for those viruses in that specimen. Over here is our microbiology area, and in this area we take a variety of specimen types, whether it be cerebrospinal fluid, urine, blood, those types of things. We put them in cultures and we grow them up, looking for certain types of bacteria, viruses, or the types of fungus or things of that nature. In this area we test about 20,000 specimens per month. This is our final area where we receive our tissues and our biopsies from specialists like dermatologists, gynecologists, and others. Those biopsies are cut up, put into wax, sliced into very small pieces and placed onto slides, which are then stained and sent to pathologists to be read and diagnosed. In this area, this is what we call our cytology, histology, and electron microscopy labs, where we do 40,000 specimens a month, over 130 specialized stains, and over 40 individual procedures.
Intermountain's Central Lab is distinctive. Dr. Sterling Bennett, medical director, explains how the team here assures quality and accuracy.
Most patients never think about what goes on in the laboratory, but we think about it all the time. We know that high-quality lab testing helps physicians, nurses, and others deliver high-quality medical care. We have a comprehensive quality management program that addresses all aspects of the laboratory, in particular the 3 Ps: people, products, and practices. People are the laboratory's greatest asset. They're the main reason the laboratory works so well. We recruit and hire the very best people and invest in their training and development. Our people are committed to excellence. They give their best with every test. Products encompass our analyzers, reagents, computer systems, and other materials needed to run a laboratory. We spend a lot of time and effort finding equipment and supplies that meet our exacting standards. Selection is based first and foremost on quality, not on cost. Products also include the reports the laboratory sends to providers. We continually expand our capabilities to deliver reports quickly and accurately in the electronic or paper formats each provider requires. practices include our testing procedures, rigorous quality control, and other activities designed to produce the best possible results. Our testing procedures are defined by teams of the brightest and best laboratorians from throughout Intermountain Healthcare. We publish daily, weekly, and monthly statistical reports to help assure the accuracy and timeliness of our results. We have high-quality targets. Then our people continuously monitor our performance, identify opportunities for improvement, and revise our practices to raise our quality even higher. Here in the Central Laboratory, as in all Intermountain Labs, we constantly strive to strengthen our people, products, and practices because ultimately we are passionate about patient care.
One of the reasons this lab consistently produces quality work is the team. Let me introduce you to Tristan Duckworth, a client services rep, and one of the more than 300 employees who work here.
Our team is incredible, from our phlebotomists and our draw stations to our couriers, our client service representatives, and everyone who prepares specimens and performs tests. We all know our work is important, and we take pride in the services we provide. We know we're making a difference in our patients' lives, patients who are our friends, families, and neighbors. Everyone deserves the best, and that's what we strive for.
The quest to serve patients is universal in this lab, and the patients feel it.
I was blessed February 1st, 2004 with a beautiful little boy. Jonah is actually the only known child in the world that has 2 rare blood disorders. So, and yeah, he did have open heart, but with the blood disorders, we rely heavily on laboratory services, with just making sure that the labs are correct, and he gets so many different types of labs drawn. I just felt as a mother, it just was so nice to go somewhere that you knew that they were going to treat your child with love, that they almost looked at Jonah as their own because we saw them so much, and I just felt very confident just going in there because I knew that they would take good care of Jonah.
They only give me 1 pump. I just got one yesterday. You can't really see them, it's red, but I don't really cry a lot on my IVs. I get hyper a lot with blood because I get treats and chocolate milk.
The work in this lab goes on quietly and without fanfare each and every day. But this team is instrumental in changing the lives of hundreds of thousands of patients. On the occasion of the 5th anniversary, we salute the men and women of Intermountain's Central Lab and thank them for their commitment to excellence.
Okay, so that is the first department for our facility that I worked at. And I actually got offered a job before I even left college. And is it okay if we leave the slides off so they can see the board, or is it going to mess it up?
What's going to be best from the video? Probably, I think it's okay.
Okay, perfect. If you guys are okay, we'll keep going. So I got hired there right out of college 2 weeks before I even graduated, and started working grades. At that time, I was working 11 10-hour shifts. It's called a 7-on, 7-off schedule. And so I would go for late at night and stay there till the wee hours in the morning. And the laboratory never shuts down. I would do 4,000 specimens in a shift in one department as I would cycle through each different day that I had. There weren't very many of us on grave shift. And the next person would come in and I would tag them out and go home, and they would work, and someone else would relieve them, and then I would come back the next morning, or the next night, the morning for me, but night for them. That's how we do it. So medical laboratory sciences is what the degree is. And a lot of times they're like, hey, you work at a hospital, Quinn. Are you a doctor or a nurse? So there's a whole team of people that work to keep, whether it's respiratory therapists, so on and so forth. There's the 2 fields, medical laboratory scientists or clinical laboratory sciences is what you might find around depending on what region you're in. They've recently changed the name. Well, not recently. When I was in school is when they changed the name. Because I graduated with a clinical laboratory science degree. These 2 are basically synonymous, and this is it's a 2-year degree. And this is what we do. So when we were swapping the fluids, right, we look for the detection, diagnosis, and treatment of disease, okay? Based on what we're finding in the body and the specific analytes that we're testing, we can monitor what's happening, whether you're getting better or worse, whether the treatments are effective or not effective, right? And that's for all different types of specimens that we can receive. And the types of facilities that we can work in are a range from hospitals, clinics, research, developing new tests, and trying to validate new things or even producing things like business and stuff like that. Okay? So let me know when I can move on to this. Yeah, so most physicians aren't doing their own testing, and we have teams and teams of people that are doing this. I get up at 4:20 in the morning, so that I'm at work by 5 a.m. I actually will run analysis on the instrumentation, make sure our water's good, that the lamps and all of the actual analyzers that we do are good to go. And about a good portion of my day, nearly 50% of my day, is just focused on making sure that the analyzers are working properly and giving us good results. Because there's a lot of regulation involved with keeping things going. And it's not seen, but it takes a lot of work. I actually got a phone call in the last class I was in. They had a jam that they were trying to fix, and they called me. I'm actually working in a very specific department now in my title. Are we good to go? Okay. So the job description, once again, is we analyze samples, and this is all samples from the body. Poop, pee, CSF, blood, you name it, comes from the body, we test it, okay? And we look for different types of disease or the treatments of it. Because it's so complicated, and the different groups of information that we're trying to do, and limitations of analyzers, we have different departments. Blood bank, this is transfusion services, like if you're receiving blood. Hematology, to look at your actual white cell counts. Platelets, your red cells. We look at the size, the shape. We look at the colors that are in them. We differentiate the different pieces and parts, and we put that all into the calculation to see what's going on. Chemistry is what I do. I'm actually a lead chemist for Altitude Hospital. I do a lot of the field reports for my medical director. I'm also on the Chemistry Practice Council, or work group is what they call it now, and I help make decisions for the entire corporation. Okay. So there's some logistics stuff that is taking place with not only running patients, but to be honest, once I get things done, it's all taking care of patients' care thereafter. We have other departments such as like education. Point of care is like what the nurses use. When I worked up at Heber, there was Heber Valley Hospital was having a hard time with staffing, so I went and worked up at Heber Valley for a little while. And when there was a trauma, I was the only person working. I would work all these departments by myself alone, and I'd run out to the traumas and go to the trauma rooms and actually run the trauma labs right there, and then go work them up again when I get back to the lab with the actual good instrumentation, because there's limitations. And then at Central Lab, when I actually left, I worked there for several years. When I left there, I was actually rolling out the LCMS mass spec. And what we do there is we ionize, or we destroy the molecular structure, and based upon its mass and its electrical charge, we send it through a flow cell that's bent and use a very strong magnet. And depending on how we deflect that direction, we have a sensor or a detector that actually lets us know what's in there. So, yeah, that's how we know what types of things, or research, which is kind of cool. Okay, you guys are kind of getting glazed over a little bit. Let me take you to the UA department. So let's turn on the light and head back over to UA. One of you grab the light and we'll go over to UA. Come on, out of the sheets. I don't bite.
I think I'll go up high on this one.
Okay, are we good?
Yeah.
Okay, so welcome to UA. This is how the hospital is set up. When I go into my department, the UA department is a section of the actual laboratory. So there are actual physical departments that are split up. In some laboratories, there was a bench where at Central I would do nothing but urine analysis all day long. That's all I did, okay, because of how much volume. Whereas the hospital I work now, my colleague, she's doing hematology, body fluids, covags, and urine analysis all in the same area. So she's a rock star. So what we're going to do now is we're going to do a random drug test for all of you. That's a joke. I look for who's like, oh no. So what we do as a laboratory, okay, just so you know, if you come to the hospital and you're doing drugs, we're trying to help you. Please tell the physician so that we can help you. Don't lie about it. We can help you if we know what's going on. But the first thing that we do with drug or UA analysis is it's called a macroscopic. These strips have a chemical structure on them that will bind and react with the different things that could be potentially harmful for you in your urine. Who knows what urine is? It's pee. But where does that pee come from? From the kidneys. Where does it come from the kidneys? Yeah, but there's an intermediate step between there. So your stomach breaks down the food, absorbs it into your system, blood, and that's how it's actually fed throughout the body. So testing your urine can be a very vital way, and a simple way, because we don't have to jab you with anything, to get urine. You'll get it quite easily if possible. And we can test that and look for things that would be causing you harm, okay? So that's a kind of a simple test. And you guys will be doing this, I believe, later on this year, okay? So this is kind of how the body system works. Who has the next birthday? February? February?
February 21st.
February 21st. Can anyone be February 21st?
February 21st.
Wow.
Ooh, time.
Birthday.
Morning, afternoon?
I think I was born more towards the night.
Okay. You know? Afternoon? Okay, you're my victim. So go ahead and come up here.
You're gonna pee in front of the class?
Not exactly. So what I'm going to have you do is, I've got this little tablet, and I want you to use that, and go ahead and put that into one of these vials of specimen. Actually use this. I don't want you touching it, that's why I got the thing. So go ahead and slide it in there. Boom. Okay, awesome. Way to go. Okay.
Interesting.
So go ahead and go back. So what we're doing right now, this is a special test, okay? So what's happening is this tablet is reacting with the specimen that I have here, and it's going to make a color change. Based upon the color change, we'll know how much reducing sugar is present in this patient sample. We normally use these for infants, and here's the big deal, right? So if a newborn is born and their body can't absorb the glucose that mom was giving them, or the mom that, you know, the food that they're taking, what's going to happen to baby? Not going to be good, right? They'll start to starve and stuff like that. So what we can do is we can do this reducing sugar test, because glucose is water soluble. It's going to pass through your kidneys, through the filtration system, and it's going to come out in the urine. And that's what we're testing. Now, limitation of this, though, is that this is going to work with any reducing sugar. How many of you guys have heard of diabetes? Okay. Anyone have a family member or friend? What type?
One that you get when you get overweight. I'm not sure.
Okay, type 2. Anyone else? Type 1?
1 and 2.
1 and 2. Wow. 1 and 2. That is rough. So what's happening there? So you have diabetes mellitus, which is it means syrup. Mellitus means syrup, which also means syrup because it's sweet. So back in the old days, what they used to do with them is they would taste the urines. This is their sweet apple juice. So we don't do that anymore. Luckily, we have chemical tests to actually do that. But here's the rub. If this is apple juice, which it is, what sugar is in this? Fructose, exactly. So that's the limitation of this test. Any reducing sugar will be able to be reduced. So there's limitations. We do tests, and it's actually exothermic. I can actually feel heat coming off of this. There's limitations to the test. So we have to do the right test without the interferencing like situations and try and actually come up with a good analysis and figure things out. Cool. Go ahead and go back to your seats. Is this kind of fun?
Yeah.
Not too bad?
Sir, okay, sis.
So far, your class has had the best reactions so far.
Yeah.
I'm glad none of you passed out on me or anything. If you get queasy, please don't stand up or anything. I don't have anything super graphic, but we have some blood that we can take out. Okay. So that is the macroscopic, right? We do large analysis on it. The next step that we want to do is we actually will go microscopic. We'll concentrate on your urine and we'll look under a microscope to see what's supposed to be there. Here's a picture of red cells under a microscope. Are red cells normal in the in urine? Maybe for females, you know what I mean, during their cycles. But for the most part, should you be peeing blood? No, you should not. The filter in your kidney should actually not allow blood to pass through it. Any of you guys play sports? Yeah. Sorry, gray jacket.
Oh, basketball.
Basketball? Black hoodie?
Lacrosse.
Lacrosse, awesome.
Basketball.
Basketball. Any others?
Basketball.
Basketball. So, what can happen, like, one of those tests is like protein. If you guys, like, work out or you're runners or something like that, you can actually increase your blood pressure to the point where it's pushing, like, proteins and some things through. The glomerulus is what it's called, that filtration. But for the most part, blood shouldn't be there unless, like I said, something bad's happening. So another thing that we might be looking for is other things that shouldn't be there. On this left side is yeast, branching hyphae, which is really not good. Most commonly found in women due to the short nature of the urethra, right? And older gentlemen as well, because the ability just to fight off like opportunistic pathogens becomes more difficult as you age. It's just natural biology. On the right side, this is calcium oxalate crystals. Does anyone have or know someone who's had a crystal or kidney stone in the urine? Oh, boom.
My mom.
Mom.
And
And?
Grandpa.
Grandpa. Ooh.
Dad.
Dad?
Uncle.
Uncle. Tragic. Does anyone have a good kidney stone story? Or something horrific happening to a friend or family member? Speak up.
So it was when my grandpa was really sick and he was like going to pass away. My aunt Wright he was getting really sick. She had a kidney stone. She was gone for like the entire time that my grandpa was really sick.
Oh, that's so
She
terrible.
was like throwing up. It was really bad.
Yeah. Anyone have anything else? Okay. So I said that I work grave shift, right, at the central laboratory, and we're working these really long shifts. There were only 4 of us running the whole lab at the grave shift, so chances to go and get water and go to the bathroom weren't as frequent as I would have liked. And so lots of us actually developed kidney stones. I got 2 3-millimeter kidney stones. And what happens is in your kidneys, which you'll cover the renal system later on, your renal pelvis concentrates it, the ureters, goes to your bladder, and you got 2 of them, right? And then to your urethra. Well, I had 2 3-millimeter calcium oxalate crystals, and they cut you as they move, and then when they stop, it basically feels like all I could do was sit at the microscope and like clutch like the microscope as like I could feel them like just slicing me open. I wanted to vomit. It was honestly terrible. So not fun at all. And so they just basically say, drink lots of water. They gave me Toradol, which is like liquid ibuprofen, which is like amazing. I could drive myself home. I'm like, yeah, this is great. And then he's like, you need to be on a narcotic because buckle up, it's gonna get bad. And I'm like, I gotta do tests. And he's like, second thought, wait until you're off work. So, yeah, super fun. Okay, so now we're gonna transition away from urinalysis and let's go to microbiology, okay? How many of you have a food that you hate? You just can't stand it? Boom, what is it?
Cottage cheese.
Cottage cheese, how dare they?
it's a soup my mom makes. It's like broccoli cheese soup.
I hope she knows.
It's so bad. Horrible.
Beans? Like green beans or like Oh, beans, like kidney beans, any bean.
Anything with cilantro.
My wife is the same. She can't stand this stuff. She says it's too much.
Pretty much all green vegetables. I just don't like them.
Seafood.
Seafood, raisin.
So bad.
Well, I can't stand onion. Onion is so gross. I mean, onion is like that friend that you bring over and you're up way late and you go to sleep and then you wake up in the morning and you're still on your couch. You're like, go home, the party's over, just go. That's onion. Anyway, bacteria are the exact same way, okay? They're fastidious, they're picky eaters. So what we have here are two agar plates, okay? This is a sheep's blood agar, and what grows on this are organisms that can break down the red cell and get to the nutrition inside, right? And depending on what will grow on this, will differentiate someone. Like if we were to separate people who like pickles and those who don't like pickles, right? On this one, this is a McConkey plate, which has probably the enterics, anything in your GI tract, which obviously can cause lots of infections. And so we can separate these out. Not only will we do that, we'll put them in the best situation. A raise of hands, how many people prefer summer versus winter? So summer first, winter, see what I'm saying? So bacteria are also the same. So we'll keep them in aerobic and anaerobic situations to try and differentiate them and allow them to grow. So what we're trying to actually accomplish here, and what you want to do with your agar plates is you always set them with the lid down, so as they grow, condensation will drop onto your lid and not onto your organism. So what we'll do is, it's called isolation. We'll actually streak out the organism and try and get isolation. So what we're doing is we're sterilizing between each time that we're going through. And we're trying to isolate individual colonies, or if there's multiple, we want to isolate one individual colony. The reason why we want to do that is so that we can actually do additional testing on it. Now, the very first time that we get some sort of infection, the quickest thing we can do is a gram stain. What we will do with a gram stain is we will flood all of the bacteria with a primary stain, which is purple, crystal violet. We will add a secondary mordant, is what it's called, it's iodine. And what that will do is it'll bind together and make a giant complex. So that when we treat it with this ethyl alcohol or acetone or some sort of decolorizer, depending on the cellular structure of the organism, it will open up pores, allowing it to be washed out. That complex will just flow right out. And then we can hit it with a counter stain, safranin, and it will stain everything else red. Now we know, okay, what kind of structure we have, and we can differentiate. We can take the boys on one side, the girls on the other side. And that's the kind of idea, so we can know what we need to do. That's important because of this. After we've identified it, let me just mention this, we can actually take those isolated organisms, and we can do chemical tests as well to allow us to know. Can they, do they have catalase or do they not have catalase? And are they allowed to, able to deal with peroxides? So here we have what's called, this is a Mueller-Hinton agar plate, and also known as a Kirby-Bauer plate. What we will do, instead of trying to do an isolation, we'll do is known as a confluent lawn, and we'll spread the organism all the way out onto this plate. And then what we'll do is we'll add antibacterial discs to see if there's a zone of inhibition, right? If we put them in a situation in which the drug interferes with their reproduction, or how they eat food, or will destroy their cellular walls, or won't allow them to build them, you can see we can get effects on the different types of drugs. So, normally we don't do just one of these, we'll do several of them. When I was on my rotations at in college, I went to a micro lab that had Pseudomonas, and they probably had 20 different antibiotics on the Kirby-Bauer plate, and all of them looked like A. Nothing touched it. I was like, oh, that's super scary. What do you do with an organism that you can't treat with an antibiotic? Not a rhetorical question. Any ideas? So we have to remove the tissue that's infected, and you generally will have to take healthy tissue with it because it's just going to spread and continue to go. Debridements are another thing. They'll scrape out the wound. and really bad. If you get blood in your bacteria in your blood, it's called bacteremia. If it actually is growing in your blood, it's sepsis. Very scary, very serious situation in which, has anyone ever heard of MRSA, methicillin-resistant Staph aureus? How we get these situations is things like this. You see how F is affecting only half of it? If you've ever been given an antibiotic, oops, sorry, other screen. If you're ever given an antibiotic and it says take it for 14 days, I know you're going to feel good by like 10. Please take it the full 14, because we need to lower the bacterial load to the point where like your body can actually fight it off and finish it off. Okay. So I don't know if any of you have noticed, I've got a little bit of mustard right here on my lip. So this morning I got a cold sore. On my lip. So by being able to isolate, I know that a cold sore is an envelope virus. Okay. So vinegar will break the protein coats for the envelope virus and break it down. However, if you just rub vinegar on it, is it going to stay there? No, it's going to evaporate, and I'm here at the school trying to teach you guys. So grab some mustard, which has vinegar in it, it's the second component, and it will dry as a paste, and I can just apply it throughout my day. Now, if I were at home, I would add the actual, it's called cyclovir or Zolvirix, and I would alternate between those so that I would be destroying the protein coat and keeping it from replicating and get rid of the cold sore a lot faster. Okay? So knowing what we're doing allows us to isolate and treat diseases. Okay? Sweet. So let's go to blood. So hematology department, we look at the different blood. What time do we get out of this class?
11:10.
11:10. Perfect. We got about half an hour. So based on the blood, we have 3 different components in your blood. And I'll use this real quick. Well, that's a terrible marker.
Sorry.
Well, you guys get the idea. So right here, so we'll have, you have your red cells down here. Then there was going to be what is called a buffet coat. This is your platelets. What do platelets do? You guys know? They help with clotting. And then white blood cells, which we're discussing now. And then up here, we're going to be Smurfs today, so we're going to have Smurf plasma or serum. And that has like clotting factors and other things in it. And it actually allow us to like move this stuff. Men have a little bit higher of a hematocrit than women. It's like 40 to 36, 35. So up here we have the white cells. White cells help fight, for immunity. This is a blast. Beyond this, in your bone marrow would be a pluripotential stem cell. And then it would go to this blast, and this is in the myeloid series. So we're talking granulocyte cells. Any of you guys allergic to anything? No one's allergic to anything?
Gluten.
Gluten. That's so sad. I love bread. Anything else? There are gluten-free breads. So. Anything else? Okay, so I'm allergic to cats and dogs, so I had to stop eating them. But that's what these are for. Eosinophils help with parasites like this, like a tapeworm, or for like allergies. Down here is called a SAG or a neutrophil. These produce lysosomes and peroxisomes that actually destroy bacteria. They go up and try and dump those on there, and then basophils are really for histamine. There's macrophages, which are another thing, megakaryocyte, well, not megakaryocytes, but macrophages that actually will engulf and eat things, even other cells that are contaminated as well, to help clean things up. Any of you guys seen this really old movie? I think it came out in 2001 when I was a senior in high school, called Lord of the Rings. Anyone seen it? Maybe once? Maybe twice? Did anyone see the second one? And where there was like the Battle of Helm's Deep, but a lot of the men died on the way there, and they actually had to keep fighting the war at Helm's Deep. Who did they send out afterwards? Does anyone know? They got the little, anyone, like any of the small boys, and they put armor on them and send them out. The body is super resilient. It does the exact same thing. So when I'm doing my analysis with the blood, if all of the mature cells in the bone marrow are already out of the vasculature because you have an infection, it'll continue to send out more immature cells to try and compensate for the system, which tells us clues about what's going on in the body and how much the body is trying to fight the infection, okay? Red cells. When we do our analysis, we look at the size and the shape of the red cells. What do red cells do? Do you guys know?
Carry oxygen.
Carries oxygen, okay? The size, the shape, the cellular structure, the pigment, and the color with our stains lets us know what condition our red cells are in, okay? This helps us know what type of diseases that we have, or genetic, or things like that. So let's go into this. Here's a hematology where we have the whole series in someone's blood. This is kind of a thick area for blood, actually. We like to see it monolayered, more like this. You can see that these red cells are misshapen. This can happen with genetic disorders, or if you're struck by lightning. This can cause problems in the cell structure or electrocution. Or another thing, the spleen could be taking things out. These acanthocytes, and also known as helmet cells. These down here, however, are called spherocytes. And normally what's happening here is clotting is happening inside the body. And it's causing like Swiss cheese graters, and it's cutting the cells up, which is not good, because that will cause lots of problems in the blood system. These are lymphocytes, and lymphocytes actually produce antibodies to help us fight infections, right? I mean, if we see an antigen and we see it as foreign, we'll produce an antibody to let the body know we need to destroy that item. The problem, though, is this person's lymphocytes are big and they're abnormal. And these smudge cells here is a situation in which when you smear the blood out, it's breaking, and this person actually has CLL. This is a very treatable and not very life-threatening leukemia that you have, but this is a telltale sign of a leukemia, and these are blasts. These are really big problems. We then have to call the pathologists and the doctors and let them know that we're seeing something with leukemia. We really want to get it tested to see how aggressive it is and what ratios it's at. Down here you can see an actual nucleated red cell where the spleen hasn't removed that. So with that being said, let's actually go to hematology department right now and be super quick and have some fun with hematology. Let's go. Okay, so over here I have small, medium, and large gloves. Please get gloved up right now. Grab a set for you, grab a set for you.
JC at 30 to get a large.
Okay, so gather around. Please don't be shy. Just don't block the camera so your friends can get in here.
We got the same one. I grabbed two.
Okay, come on, gather around, gather around.
I like opening you guys.
Okay, so what I want to do.
Problems.
Okay, so this is my blood. I had it drawn 2 days ago up at the university. Okay, one of my friends, she was nice enough to do that. Some of my slides that I brought have this frosting on it. Okay, that's so that we can write down information. Because if I took your blood and resulted it on you, and we started treating you on it, that would be bad, right?
Yes.
So identification is super key. And I have to trust the people who are drawing the blood, both nurses and phlebotomists, that they're doing a good job, okay? So what we're going to do is, this is a helicopter top. You're going to use pressure on this helicopter top, turn it directly upside down, and you want to go to the back side of this frosted edge. If you have a frosted slide, your non-dominant hand will anchor and put pressure on that and push down, and you're going to put a very light drop. Smaller is better, okay? On there. I want you to hold this between your fingers, not like a credit card. Don't spend money. Hold it like this, okay? You're going to hold this firm. You're going to contact the top of the slide, and you see how the blood is back by my hand, like this. What I want you to do with this is you're going to go back and forth. You should feel even pressure, okay, all the way across the slide. You can practice this a couple of times, okay? And what we want to do is we're going to touch the blood,
blood on the back side and then run it forward nice and smooth, okay?
If you decrease the angle of your slide, it's going to run long. If you increase it, it's going to run short, okay? So I'll demonstrate this. Kind of like, have you guys driven a car? Have you ever backed up and bumped into like the garbage can? You're like, oh!
I thought I was a kid.
Okay. That's unfortunate. Then they would end up in my hospital. Okay, so here's I'm gonna show you how to do it. Okay, so you're gonna come back here. You're gonna touch. It's gonna wick across, and then forward just like that. Okay. You don't want to lift up. You want to let it slide right across, and it's gonna make that thumbprint. That's a feathered edge. Okay. Okay, everyone get 2 slides, and let's go ahead and do it. We've got 2 over here, and I've got more over there, and I will help you. Come. I'll grab some people. Come over to this side.
What side? Doesn't matter what.
No worries, we'll just carve it.
And what you do, find the front side, okay? You're going to anchor with this finger. You're going to hold this one firm, and I want you to set it on there and go back and forth like this. Just get that feeling. Because you don't want to lift it up, you don't want it to rock. Okay? Nice and smooth. So your drop of blood will be right there, your drop of blood will be right there, your drop of blood would be right there, okay? Closest to your finger, okay? Okay, so what we're going to do is you're going to drop your blood there, nice and small.
Just to
And you're going to come back.
Now, what side do I use?
Go ahead and try. Okay?
Oh my gosh,
That ain't no thumb.
Okay, just so you know, this took me an entire semester to master. It's hard. It's supposed to be, okay? So you're going to be anchoring on this side. If you don't, are you right-handed or left-handed? Okay, so wind up your finger.
Dude, that was fire. Look at that.
That is not.
My side?
Yeah, Finny?
That's what you gotta do. Okay.
Dude, it's just like frosting the cake,
Do
bro.
it once.
You were backwards, but that's okay. So go ahead and touch it right like that. And that's why I like holding it like that. So you can hold it like a credit card. Come forward.
Yeah.
Here, let me grab your slide.
That's the best.
I'm gonna show you one more time.
We
That's my
ignore
close
that part and look
Okay.
at that one. That one's 10 of it.
So touch it right here, just like that. Okay, so I anchor it. See how I'm drawing on the back side, coming back like this. You touch the blood, come forward. Okay, if we were to actually stain these, they would no longer be biological specimens, and you could take them home and put them on your fridge. Your parents would cry about how amazing you guys are. But since they are not stained, we're going to actually put them in this biohazard bin, and I actually want to show you how to take your gloves off. You're going to grab one glove, turn it inside out, wad up your hands like this, and you're going to turn the next one inside out and discard it, okay?
Once you guys have had a chance to try it, head back and let the second group come in.
Over here.
Wait, no, I don't do that.
Wait, I want to let my microscope. He said take it off the table.
Was this kind of fun?
Yeah, dog.
Not too shabby?
Oh, the second group didn't even want to do it?
Oh, y'all are pansies. Oh, man.
Just told the first group to move.
So go ahead and grab that inside of that one. Okay, and like I said, we were trying to produce that really thin layer so that we can see what's going on and differentiate each of those cells. So quickly, does anyone like to do anything dangerous?
Yeah.
Give me an example. Skiing? Motorcycles? Okay. I'm a motorcycle man myself. So blood bank, if I were to get hurt and start bleeding, blood bank is the way they would actually get blood cells back in your body, okay? So we actually separate out, and they're called pack units, and we actually will make type-specific blood. You guys will do this later on, okay? So if I were to get hurt and start bleeding, they can put red cells back into me. However, there's situations like a trauma, or a situation like there's a situation with a pregnant mom, delivered, she started bleeding. She started bleeding, and then bleeding, and then bleeding, and bleeding. And we put 18 units in her. At that point, she was no longer bleeding out her own blood, right? We completely replaced her blood volume. So, what you have to do then is actually give clotting factors and platelets, because she's let all of these things out. You know what I mean? Super big deal. And this is a life-saving technique that we can do to do that. And I believe you guys are going to do your own blood typing later on this year. So super, super cool. Because if you give someone plasma to the wrong blood type, you actually will attack those cells, see them as foreign, and it will lyse them. And now you're bleeding to death, and then you've got the hemoglobin and the blood pressure dropping, and it's just really, really bad. It's a bad situation. Education. So I went up to university, Weber State University. You can actually go up there and you take 2 classes as a prerequisite, basically, to try to get into the program, and you can become, it's called a CLA when I went to school. This allows you to do like phlebotomy and do other processes, plate things, deal with specimen processing. Not always do you have to do this though. You can go up to the hospital and actually they can teach you how to do these things. You can do classes and actually start working right immediately for the hospital, which is super important. Because one of the things is some of the hospitals will actually do a reimbursement and help you pay for college, which is awesome. And just so you know, concurrent enrollment, AP classes, anything that you can do to keep paying off your student loans or not have any is super smart, I'm just telling you. And hopefully by now you guys already know how you learn, okay? The biggest thing I can tell you is learn how you learn. Like I said, I was an artist, and so I spent a lot of my time when I was taking a heavy credit load. I couldn't do all the homework. So what I did is I would draw the theory in my own pictures, and I would study my drawings, and I could do more with that than all the people doing the lists of notes. And I got through college, it was fun. I'm the most average student you'll ever find. I was a kind of a B average student, and I mean, her class was kind of hard, and you know, made through it. So after you apply to the program, you have a 2-year, where we do all these different fields we just talked about, okay? You do a 2-year associate's degree. You can actually go work in the field after you take an accreditation test for those 2 years and get your MLT, okay? That allows you to actually work in the field. You can then apply again for the program, for a 4-year degree program. It allows you to go a little more in depth, and also these more complicated analysis that you have to do. It allows you to do that, or do supervisor, or go on to other different departments, if that makes sense. So it just opens some doors for you, okay? Weber State University has a simulation lab. That means as I was going through school, I was doing these tests and these fields very hands-on while I was going through. And I didn't have to deal with any patients. You know what I mean? Not listening to me. My wife's a terrible patient. She's a horrible patient. I'm just teasing. Don't tell her that, but it's true. Research, my senior year, I got to do a research project. I could have published had I had a little more than the $45,000 that we were allowed, because of how expensive the research was. Also, my rotations for the hospitals, once I was going into the rotations, it was very short, 2 weeks for each individual department that I was doing. Versus University of Utah and Brigham Young University, you do 2 years, your junior and senior year, you just hit the books for nothing but that. And then you do a 6-month human slave where I get to work you to death at the hospital. You know what I mean? So both of them work, it's just whatever works best for you. They also have online, so if you're working in a hospital, you can actually do your rotations as you kind of go through everything, and they can get that all set up, if that makes sense, so as you're doing your schoolwork. So there were prerequisites. It took me 5 years to get my 4-year degree, ironically enough. A lot of you have a lot of opportunities, like I said, with these concurrent enrollments to actually get college credits. So you can graduate with your associate's degree already ready to go. Whereas I had to do those classes. I was an art student, so I was like, I don't need any of this stuff. And so education will only open doors. It will never close them. Just to let you guys know, okay? The labs were really long. Sometimes I would be there from 7 until 7 because there were 3-hour labs or 2-hour labs or long exams. Our exams, however, were both laboratory and theory. that we do, and sometimes even practical. We have to demonstrate the ability to do the procedure or the function that we're doing. You had to pass with 80% or better. Who wants a C or a D person looking at their blood? No one, right? Because it's a big deal, right? If I miss cancer, if I think that you have a heart attack and don't, how about if I think you have a horrific disease and we pour a bunch of poison in your body? Big deal, right? So 80% or better. You fail a test, you get to retake that test or a similar test of that type. If you fail it again, they give you one last shot. If you fail that one without an 80% or better, go pick a new major. I was in my senior year doing my one of my last chemistry exams, and there was a spreadsheet where I did a lot of calculations, and I remembered at the very end I was like, oh yeah, you wanted me to express this in a percent, so I said set express percent, but I'd actually built percent in the formula, so it multiplied everything by 100 again. 0 on the test. Luckily, I just went back in and I got to change those answers and just take that application off, and then resubmitted it and got 100, but I only got 50 points out of that test because we take the average, the score. It cost me a whole letter grade. I would have graduated with honors. It's okay. But I'm just saying, like, you know what I mean? This is kind of like a big deal. I am the most average student, so don't let me like scare you with this. You can do hard things. All of you can do hard things, okay? So, after you're done doing your education, you will then take an ASCP board certification test, basically, and for the clinical pathologist. It's one of those, if you get the test question right, it's going to ask you a harder question. If you get that one wrong, it'll ask you an easier question. So it'll get harder and harder the better you do. So if you're sitting in this, I think it was only a 4-hour test, and you're like sitting there and you're like, oh no, I'm going to fail, you're probably doing pretty good. You should feel like you're about to fail, because that means that you're doing a really good job. Actually, I had a colleague who his goal was to get 100% on that test. Good luck, Scott, because he probably did it. He was super smart. It was hard. Continue education. Every 3 years I have to renew that. So I actually just submitted and got my renewal for my accreditation this month, and I actually have to send that to my work to show that I'm good for the next 2 years. Okay? And that's for all the different departments, so that the breadth of knowledge that we have is really awesome. Okay, earnings. So like I said, as I was going through school, you could take those first 2 classes, and I had friends who actually went in and became laboratory assistants and phlebotomists and specimen processors, and started making really good money while they were going to school. And then they also got tuition reimbursement, like I was saying. After we finished that CLA class, I had colleagues who actually would work in the laboratory, you know what I mean, specifically, just helping out around there. I actually got my 2-year degree, and no one would hire me because I was too close to being close to my bachelor's degree. But I bounced around and did toxicology and worked for the actual lab at the university while I was going to school, which helped me make extra money. I would do samples, much like the simulation that we're doing today for the incoming freshmen. And then I actually had offers for my bachelor's degree before I even graduated. Okay. Now you'll see there, all of these values are for the state of Utah. Okay. We run lower than the national average, just so you know, and we also, so you might see higher wages. Down there at the very bottom for the bachelor's, I have someone with 41.20. I have a colleague who's been doing this for about 20 years, and he's making about $40 an hour. He's making some serious bank. And then with overtime, that's time and a half, so he's making about 60 on the time and a half. He does it. Okay. What else? I don't want to go too quickly. Does everyone have this information? Are we good? Anyone say no? I'm in your way. I'm so sorry. Okay. Any questions, concerns so far?
So how easy is it to find a job in these fields?
Right now, it's quite easy. There's always turnover, and we'll kind of get to that in a little bit. With that being said, I just want to make sure everyone has time to write. So, with finding a job, with this degree, you can kind of go on. Supervisors, managers, and directors can make more. So you have people who are always leaving, you know what I mean, on normal work, going into those other fields. We also have, the University of Utah right now has a 1 year, like, it's just by contract for $95,000 to go be an adjunct professor up there. Super awesome, right? Go and make 95. The Dean, Yasmin Simoni, teaches, actually is the Dean of the College of Allied Health up there. She's making over 6 figures going back into education. Now, a lot of these people will go back and get their master's degree in something for their education or whatnot to do so, okay? But what I did is they actually have 2 tracks. There's a normal medical laboratory scientist track, or there's a pre-professions track, okay? And that's what I did. I was going to go to dental school because I knew that I wanted to blend art and science together, okay? So I did all of my medical laboratory scientist stuff and the stuff for pre-med, and pre-dental, and PA, and all that stuff.